I would like to, I'm very grateful actually for the presence of Dr. Marensi. Dr. Marensi is going to be part of our meeting as to push a little bit our international collaborations. So she will introduce herself a little bit later, but in the end of the day, this meeting is mostly related to try to connect some different research areas, actually somehow in the same field, but research topics, trying to make connections with collaborators from abroad. And in this sense, I'm also grateful for the presence of our colleagues, Professor Nara, Professor Vinicius, Professor Tania, who will take part in our meeting, sharing a little bit of their results or their research areas, actually, regarding pharmaceutical sciences. So, and I'm also grateful for the presence of other researchers who are attending this meeting and also students in different levels, graduation or undergraduation students. First of all, we will have our presentation from our research groups. So we can just let me share my presentation actually. So we can start giving an input on the research we make here in Univali. And later, Professor Marensi is going to share her presentation regarding her research area in UK. So I will start the presentation. Please, if you have any doubts, any comments on the ongoing presentation, please feel free to interrupt and ask the questions. But of course, we will have a bigger discussion at the end of the session. So my name is Luiz, and I'm a member of the chemical area of our research groups. So we are all in the pharmaceutical sciences department, so we will always share some common interests, but my interest is mainly related to the chemical area. And I'm going to share in the name, of course, of the research group, our studies regarding natural products and regarding synthesis. Just to be clear, I'm not a synthetic chemist, so I'm just going to show you what other professors in our field are doing, but I cannot give further input regarding this specific topic. But of course, if we detect some connections, we can have another meeting to further discuss something related to the synthetic products. So the first group I'm going to give some input is on bioactive natural products. And this group, as you can see here, it's led by four researchers. Professor Sequinel Filho, who is our rector nowadays, Professor Rivaldo Niero, Professor Angela Medeiros, and myself. This research group, of course, as the name is already stating, we work on the field of natural products. Some studies related to the quality control of herbal medicinal products, but this is always in collaboration with Professor Tania or her group, as you will further see. But our main focus is on the isolation of bioactive natural products. And in this sense, we have different approaches where we can which we can use to reach these goals. We use two different approaches. The first one is a classical approach, which is the bioguided approach, where we work with a very complex mixture, such as an extract, plant extract. We mainly work with plants. So we started with the plant extract, and we evaluate the pharmacological activity of this plant extract, and then we can chemically obtain fractions of this extract, leading to less complex samples in order to try to identify the main compound or the active principle responsible for the pharmacological activity. We use different approaches. We can use the classical approach, we're using TLC and classical column chromatography, but we also can use some other methods such as the replication by LCMS and targeted isolation by flash chromatography and etc. So here I have some references where we use this approach to obtain some bioactive compounds. And of course, we will share this presentation with you. And if you can, then you can take a further look at this reference if you want. Of course, these have some drawbacks because we know that extracts and complex mixtures, they are not usually high throughput screened. So we have some drawbacks in this approach, but we can have some success using this bioguided assay approach. Sorry. And another method we are using is the metabolic profile approach, where we use different samples, usually collected in different regions of our country. And this leads to a chemical variation, and we can correlate this chemical variation to the pharmacological activity by mathematics, multivariate data analysis, and using biochemometric approach. So in this sense, we can highlight some compounds that can be bioactive or can somehow be related to classification of the samples. So we can target isolate these compounds. And we've been using this approach for some years now, and we have some also references. I'm listening here related to this approach. So in the end of the day, both approaches, they will lead to the same end, but using different techniques. Here are some ongoing projects we have. We have some collaborations with South Korea, with Belgium, Greece, and also different fundings from our projects approved in the different agencies, also from Brazil and abroad. So also we will share, you can take further look, and if you have any questions, of course, you can ask us. Okay. And this is a quick overview of our natural products research group. And I'm going now for the pharmaceutical chemistry group. I'm also a member in this group. So this group is, we have 5 professors: Professor Sequinel Filho, Professor Rogério Correia, Professor Fátima de Campos Buzzi, Professor Rivaldo Niero, and myself. And we have 2 main focuses. One is related to the synthesis of new compounds or semi-synthesis and their evaluation, always in collaboration with pharmacological groups, and also structure-activity relationship of synthetic and natural products. And as I mentioned, I do not work with synthesis, so I do not work in the first topic. I just work in the second topic, mostly related to in silico approaches. This is a slide that Professor Fátima shared with us, where she tried to summarize some of the compounds they are working currently in this group. And here you can see different scaffolds and also semi-synthetic derivatives from natural products. This is also something they also work in the pharmaceutical chemistry group. For the in silico-based approach, we also work both in the structure-based virtual screening, mostly molecular docking, and also we work with ligand-based virtual screening, also, once again with biochemometrics on the exploration of the chemical space. And this is where I try to work a little bit. Here are also some references I'm sharing with you where you can find further information regarding these publications related to these approaches, both structure-based and ligand-based. So we can further discuss these in silico approaches. These are some ongoing projects we have approved. I have one in collaboration with Belgium, and also Professor Fátima and Professor Rogério has some. They also have some projects in this research area. So very quickly, very briefly, I'm grateful for your attention, and we can further, of course, discuss specific topics. I can take my presentation back if you want later, but I think this is just to give an input on the research we are currently doing in these groups at Univale. Okay, so thank you very much. And now if Professor Tania can share her presentation, I'll just skip my presentation.
Okay, good morning. Very nice to see you again, Dr. Vanessa. I will present in the research group of pharmaceutical technology and quality assurance. Very briefly also, this group has 6 components: Professor Clovis, Professor Daisy, Professor Angélica, Professor Josiane, Professor Ruth, and myself. We have 5 research lines concerning the application of biopolymers, development of herbal medicines, Professor Ruth, especially with development of dosage forms, controlled drug release systems. We also work with validation of analytical methods and research and development of cosmetic products, especially Professor Daisy. So I will resume the projects in development and actually in each line. In the first line, we have projects concerning microencapsulation of natural products for cosmetic applications, and also for bioadhesive systems and modified release therapeutic systems for buccal and oral drug administration by Professor Clovis and Professor Ruth. In the second line, we are obtaining and characterizing herbal extracts from pepper in collaboration with Professor Angela Malheiros. Also, we have projects in Itajaí. Professor Angélica works with Life Pharmacy, herbal formulations, and I have a project in partnership with the Company of Agricultural Research of Santa Catarina, EPAGRE. We are studying pigmented orange cultivated in Santa Catarina. This variety of orange is rich in lycopene, and the lycopene, being a lipophilic substance, is concentrated in the peel, in the pulp of the orange. So it's a waste of juice industry, and we are working with this pulp, and after freeze drying, we will obtain enriched extract of this kind of waste, aiming to development of nutritional supplements or cosmetical formulations based on green chemistry, also using green solvents. In the third line, especially Professor Ruth has projects in development of solid lipid nanoparticles, incorporating essential oils as antiseptic ingredients, or also nanoparticles, nanoemulsion for dermatologic products, also matrix tablets as drug carrier. And she is working also with self-emulsion drug delivery systems as carrier to nasal and topical administration. In the fourth line, we have projects in partnership with industrial pharmacies. As example, the project of Aleurites mollicanus is an Asian tree cultivated in Brazil, and from the leaves of Aleurites molucanus, we develop, it's a very long time we work with this project. We have developed dried extracts, and we isolated the markers for quality control. We development tablets, creams for dermatological application, and also non-emulsion in oral suspension for oral administration of this herbal medicine that is very that has very significant anti-inflammatory and analgesic effects. In the fifth line, especially Professor Daisy is working with waste of industry or of food, like the pinhão bark. Do you know? Araucaria angustifolia is a native tree, and the fruit of Araucaria angustifolia, especially the bark, is rich in polyphenols, and Professor Daisy is developing projects especially concerning the problem of hair print, aiming to obtaining dyes from this food waste in hair strands. And she also works with other residues like shellfish shells as alternative exfoliant in facial cosmetic products. And also, Professor Daisy is working with coffee grounds, and Professor Ruth is working with cosmetic, especially with natural activities with the pigment and regenerative activity from the peel of jabuticaba. We are also doing clinical studies to prove the efficacy and the security of cosmetic products. We have a clinical study with ozonated olive oil in partnership with a local industry, Filozon, and we are studying the stability, efficacy of ozonated cosmetics. Our infrastructure has some equipments like spray drying, nanosizer, HPLC with MS, NMS, and UV detectors, texture analyzer, viscometers, and also the Hansel vertical diffusion cell to study the permeation of the different substance in topical formulation. We have partnership with some companies, national companies actually, like pharmaceutical industry. Filozon is the local industry that is actually we are doing many projects with ozonated oil, Duas Rodas food ingredients, Zooprofit animal nutrition, Sabinza. Professor Daisy has a partnership with Sabinza to doing clinical studies with cosmetic products. So I am grateful for your attention in this brief presentation. Thank you for all.
Good morning. Good morning, everyone. I think I can continue the presentations here. Nice to meet you again, Vanessa. I will share my screen just a minute. Okay. My name is Nara Quintão. I work in the post-graduation program in pharmaceutical science. And now I will present the group of biological activity. Our research group is involved in different areas, including microbiology, pain and inflammation, central nervous system diseases, gastrointestinal system, cardiovascular and renal system, and toxicology. Of course, they have a large insertion in different projects, but I will show you briefly their main field, and I've included their CV link for great access to their research. Okay, so you can check there if they work with other things added to what we will show you. So the field of microbiology is explored by Professor Alexandre Bella Cruz and Milena Menegasso. They investigate the effects of extract isolated and synthetic substances with antibacterial, antifungal, and antiparasitic activity. And among other approaches, they also works with the analysis of natural and synthetic compounds on parasitic diseases, including immunoparasitology of neglected diseases. Pain and inflammation is evaluated by myself, Nara Quintão, and Professor José Roberto Santin. My main field is the studying pharmacology and investigation of natural products and isolated compounds to treat acute and chronic pain, including neuropathic pain, fibromyalgia, cancer pain, and chemotherapy-induced neuropathy, and mainly neuroinflammation involved in other approaches, including metabolic syndromes. And Professor José Roberto Santin analyzes the natural products and isolated compounds to treat inflammation, focused on neutrophil and macrophage functions, mainly the role in the inflammation resolution. We also work with the preclinical studies focused on 3 Rs, trying to reduce, refine, and replace animal models in this research. The central nervous system diseases are explored by Professor Márcia Maria de Souza and Professor Vinicius Gadotti, who will present his field next to me. And they work investigating natural and synthetic substances to treat neurodegenerative diseases, including Alzheimer and Parkinson, some preclinical models of neuropsychiatric diseases, including anxiety, sleep disorders, epilepsy, depression, and autism, in rodents, and also they worked with some models in zebrafish. And Professor Vinicius Gadotti also investigated the molecular mechanisms involved in comorbidities associated with anxiety disorders and major depression, including peripheral inflammation, chronic stress, obesity, and chronic pain. Professor Thais Boing led the experiments involving gastrointestinal system. She investigates the extract, fraction, and isolated compounds obtained from plant species in ulcer gastric and gastric ulcer and intestinal inflammation. She performs different approaches of different models of ulcers, including NS, AID, and ethanol-induced ulcer, pylorus ligation model, acetic acid-induced ulcer, and other intestinal inflammation, including intestinal mucositis and intestinal colitis. The cardiovascular and renal system is explored by the lab of Professor Priscila de Souza. They investigate natural products and isolated compounds to treat systemic arterial hypertension, hypertensive nephropathy, acute myocardial infarction, sepsis, nephrotoxicity, cardiotoxicity, kidney stones, using many different approaches in their lab. And eventually, Professor José Roberto Santin also studies the toxicology of many substances, mainly isolated substances, using in silico prediction of toxicity and in vivo and in vitro protocols according to the OECD protocols. Based on those fixed procedures, up and down, repeated doses, 28 or 90 days repeated doses, micronucleus assays, in vitro hepatotoxicity studies using HepG2 cells, and cytotoxicity assays. Here are some of international groups that keeps collaboration with our team. Most of them are located in Canada, United States, and Europe. And so you can check in their CV the studies and projects that they keep with these groups and the funding agency involved in these areas. Many of our researchers, as I told you, works in different areas, so you can check in their CV to better explore the research. Okay, thank you so much for the opportunity to present our team, and we really hope for a new collaboration. Thank you so much.
How are you, everyone? Thank you very much. So first of all, I would like to thank you very much, my colleagues, for the invitation and the opportunity of present a little bit of the research program that I wish to implement here in the University of the Vale do Itajaí, Univale. So I wish to let you know and that I'm a recent hired professor here within the institution. So I'm just starting my independent research group within Univale, and it's a great honor for me to be working here because that's the institution where I got my pharmacy degree. So I need to acknowledge before I start speaking a little bit about the central nervous system research programs. I need to recognize and acknowledge Professor Marcia Maria de Souza, who is leading the research into the central nervous system here in the institution, right? So a little background about myself, so I can, let me just maybe, so I'm not being able, let me change this screen. Right. Okay, so I'm just talking about a little bit about myself initially, my background. I'm a pharmacist, so I got my degree in pharmacy here at Univale in 2002. And then I did my master's and PhD in neuroscience at the Federal University of Santa Catarina. And then I went to the University of Calgary in Canada for a 2-year postdoctoral position. But then 2 years became 3, 3 became 4, 4 became 5, and I ended up doing 6+ years of postdoc within the Department of Physiology and Pharmacology of the University of Calgary, where I became an adjunct research assistant professor in 2015, position that I held until the beginning of this year, when I officially resigned from the University of Calgary. So despite all of that, I came to the university. And again, for me, it's a huge honor to be part of the research programs here in the university, where I got my graduation degree, my undergrad degree, sorry, and where everything started for me. Like that's where I started doing science. So nowadays, I'm a current professor with the School of Health Science and also a member of the graduate program in pharmaceutical sciences, right? So I started my position here in August of 2023, but I still keep a research associate position that doesn't involve any salary, any pension plan, any medical benefits or anything. But I still have the link and ties with the Department of Physiology and Pharmacology with the University of Calgary. And also in Calgary, I'm a member and part of a pharmaceutical startup where I'm a co-inventor, a chief scientist, and shareholder of a startup pharmaceutical company named Zynodyne Therapeutics, where we aim to develop safer and more efficient medication. So before talking a little bit more specifically about my research field, I would like to present to you here, because my background involves in vivo neuropharmacology, I need to bring to you all, and I'm sure that you all understand that, the importance of rodents in science. So it's just a phenomenal cartoon that I came across a few days ago showing the importance of the use of animal models, demonstrating that, most of the research in the whole world uses animal models in several different areas, like not only central nervous system, but like development of vaccines, leukemia, cancers, malaria, basically all peripheral organs research involve the use of animal. And there are several Nobel Prizes that were related to research that was based on animal work. So I'm just saying a little bit about that because my main background is the in vivo neuropsychopharmacology. So, but not only working with behavioral models, and especially when I went to Canada, where I spent 14 plus years within the University of Calgary, I was able to associate the behavioral models with more technological and advanced research techniques, such as optogenetics, fMRI in animals, in live mice, intravital imaging, and also patch clamp, not only current cell, wholesale voltage clamp, in hex cells expressing a specific plasmid, but also I had the opportunity to be part of projects involving patch clamp, wholesale current clamp, in this case, of hippocampal slice out of mouse brain. Despite I don't have the expertise of patch brain slices, but I have a background patching hex cells expressing specific plasmids, especially calcium channel blockers. So my main research then, what I really wish to implement, and we are already in the process of starting animal and cell culture experimentation in partnership with different professors here at the university. For example, Professor Nara and Professor José Santin. Is initially I'm very passionate and interested about understanding the molecular and the neuroimmunological mechanisms that are related to comorbidities that follow anxiodepressive states. So I'm also, as a neuroscientist, and through the use of optogenetic technique, which is like basically the main technique used in neuroscience, where we can stimulate or inhibit specific brain circuits by expressing the rhodopsin proteins into specific neural connections. I'm very interested about investigating and deciphering brain connectivities involved in those comorbidities, especially as associated to anxiety and major depression. So in partnership with the University of Calgary and also other universities like the Maguire University in Australia and the University of Montana, I've been working for many years, and I have several papers in the past 10 years doing screening of cannabinoid molecules, molecules that are synthetic, organic, extracted from plants like cannabis, or even endogenous cannabinoids, and also T-type channel blockers as new pharmacophores with anticonvulsant and analgesic activities as well. So I'm also interested in the role of using T-type channel modulators as a novel class of medication for epilepsy and chronic pain. So I'm going to show you some of my most important collaborators around the world, which are Dr. Gerald Zantoni and Dr. Christophe Altier from the University of Calgary. Both of them are going to be here in October for our pharmacology conference, and I really wish them to visit our institution and our capabilities. And then we have Professor Chris Bladen from the Macquarie University. He's also assistant professor within the University of Calgary. And we have Dr. Felipe Diaz within the University of Montana and the startup company known Dermaxon. So despite having some collaborators all around the globe, we are always wishing to expand our collaborators. And I'm very glad and delighted to join research with other groups. So I think that's the main basis of growing your research is collaboration. So that's what we do. Okay, so I really wish to thank you all for enjoying and watching. And I'm available if anybody wants to chat later on. So thank you very much, and you all have a nice day.
Okay, so I think I should start by thanking you all. It was a very good. I actually didn't expect to see so many interesting projects going on. And I'm also very pleased to see that many of my mentors are still at the university because my bachelor's was at Univalic, as I guess all of you. And then from that, I moved to Italy and I've done my master's in bioinformatics, which there was a lot of neuroscience involved. It was basically bioinformatics, but because we worked with signals, we ended up using a lot of behavioral models. I spent 3 months in GlaxoSmithKline, where they've shown all the tools and experiments they would do at the large scale. Now they closed the center, so I think that the industry is not really adopting the neuroscience much and is leaving to the universities and giving support to the universities, at least in Europe. And then after that, like Vinicius, I went to Canada and I've done my PhD, very long, 6 years, but I was in Edmonton, just next door. And not as lucky as Vinicius because he was very near the Rocky Mountains, whereas I had to do very long 4 hours to reach the mountains. But it was a very good experience, and I still maintain the connections with all the people that I met, and they mentored me and taught me a lot. And then, so in the title I think I'm using the term biochemical pharmacology because my major focus is drug resistance. But to achieve that result, I use a lot of chemical biology, biochemistry, so a lot of in vitro assays, a lot of cell assays. And so it's basically physiology. And I think this is the best way to put it. So because we talk about collaborating, I think the vision and the goal of being here today is to develop an international and collaborative research. And then, as you see, I maintained contact with many people that have been in touch for all these years. Just quick overview. I'll introduce you some of the people that give me structure for much of the thing I do, and then talk about the research, show you some of the collaborators, because if we ever collaborate, we might be sharing that same collaboration and be interacting. And then some of the events have been engaged, and then we can then have a debate and maybe a conversation about the things that we can do together. So I work at the universities at Chester, and I'm now a senior lecturer. I'm also an honorary lecturer at the University of Liverpool, where I've done my postdoc for 6 years. At the University of Chester, Professor Elizabeth has been my line manager and my mentor, and the one that gives me support for everything I do in terms of research. So she supports any of the projects, any of the collaborations I've been developing. And whereas at Liverpool, I have Professor David McEwen and Patrick Ayres. So McEwen is more into pharmacology, whereas Patrick Ayres is more into biochemistry. And I have included a photo of Rangan because Dave's now one of the authors, and I thought you would like to know it because it's been a Bible, at least for all pharmacologists and pharmacists. And then I have lab space in either lab. So I have some space at Chester, and I have my lab space either in Dave's or Patrick Ayres' lab. So those are all the instruments I would have available. So in terms of research strategy, as I was talking to you, is about biochemical pharmacology. And I really like focusing into acute myeloid leukemia. I really like how these cells behave, how they adapt, and how they can transform. And especially in cancer, they are very rapid. They have very rapid response to the environment. So it becomes a very interesting model to study. And I'm just going to show you focus a little bit in the techniques in the next slide. And then again, I do study drug resistance and drug discovery. And with that, I mean trying to understand how the cells, they adapt to the treatments we provide to patients, and then what are the options they have. And then with that, we want to discover new therapies and therapies that can be applied in the clinic sooner than later, because clinical trials can take time. So here, in terms of research strategy, one of the things I use is CRISPR, which is a gene editing strategy. So instead of overexpressing a protein, I believe that most of you know the technique, but instead of overexpressing a protein in a cell, you can edit the DNA in a way that you can ask the DNA to either transcribe or not a certain protein, and however you like. There are like several steps. It's a protocol that requires a lot of control, but it's very accurate. And then with that, we can create some models, cellular models. I work with AML model, and I also work with melanoma model, because AML is suspension cells. So it doesn't really give you the structure that would give you a melanoma, that gives you the tissue structure. The cells can get polarized, and then it's easier to understand how the proteins will move inside the cells to control signaling. A lot of the experiments I do, like RNA-seq, I just collaborate with companies that they run the experiments for me. I prepare the samples, they run the experiments, and then I analyze myself. In terms of drug discovery, it's a little bit of what you do, but I don't do the chemistry part. I don't do extraction. I just work with the molecules, trying to identify function, what their function would be and about it. But I think that what I do that might be particularly different is because I try to look at the molecular level. So which proteins the drug will target, how, and explain the whole mechanistic of the binding. And then a second approach, which I find very important and interesting, is drug repurposing. So trying to use the drugs that are available in the market for different use. So that would speed up a lot approval by the FDA. One of the examples is to work with drugs that act in calcium channels, to use with drugs that target protein kinases, which is the main family of protein that I work with. And then animal-free. So I'm not against the use of mice, but I have the benefit of not need to use them. I would if I had to do some hormonal studies or any other thing. Molecular modeling, I do at a limit. I use more for visualization and then to get proper coding and and analysis. I collaborate, and then I'll show you the people I collaborate with. And then I'm also interested in glycans and lipids. So I've done my PhD with lipids, basically. And then I was working with post-translational modification of proteins by lipids. I worked with a protein called GSTPI. And then, and this article is being just accepted for publication, and I can share that with you even before if you ever want. Glycans, my major interest is to understand how they regulate protein to reach the cell surface, because many of the times it's not just about whether the protein is being expressed or not, but how they are being expressed, which shape they are taking. Is the epitope site exposed to ligand? So those are all questions that can be answered and analyzed according to the type of either sugar or lipids you have bound to the protein. In terms of techniques, I use, and I actually didn't include in the next slide, but I collaborate with people that work with omics, proteomics. I will show you who I collaborate with at the protein level, but I work with a group in Boston to analyze glycans and lipids. And that's what I've done for the lipidation of GSTPI, which was my PhD project. So I also supervise several students from undergraduate to PhD students. At the undergraduate level, just to have an idea of how the projects are distributed, we just do some drug screen. So I tend to work mostly with repurposing drugs, where I just use cell models available, and then we try to understand different combination of drugs according to literature. We create our hypothesis based on preliminary information, and then we give it for them to test. When the student has a lot of hands-on, we can use those 6 months to try to create some cell model. No, we don't really do editing, but just overexpression, because there's no really time for that. And then in terms of genetic engineering, that's where we can start thinking about using CRISPR. So we can actually edit cells. We can work at the level with master students. There are other projects that we do, but this is just a general concept. And then it also depends on the type of cells we use, because some cells can be very difficult to transfect and manipulate. Some others are really easy. So it depends on the type of cells and the project that the student will be working on. And then in terms of cancer evolution, I've been using AML as a model. So AML is quite heterogeneous because it relies on the different set of mutations. And then that gives a different phenotype, which makes it harder to determine which treatment the patient is going to take. And then it's even harder because it's an acute manifestation. So the doctor needs to determine the treatment as fast as possible. So what they would do is just like initially give chemotherapy and start all the diagnostic tests that are available, and then try to improve therapy or make it more targeted to specific patients. The thing is that we try to understand the timing of these mutations. So we develop cell models with drugging. So we give a little bit of drug pressure, but then we collect different time points, and then we analyze with RNA-seq and several other techniques. Some of them, we rely a lot on protein markers as well to follow all the cell development. And I think this is one of the greatest models to study evolution. So just another thing, in terms of students, I'm now supervising some master students, which are MSc. So half of them are medical doctors from different places. So we work on data analysis and trying to learn how to extract the information from the clinic in a way that we create information that can be used in the lab and investigate in vitro and try to translate back to the clinic. In terms of masters of research, we have this difference here. then we go to the lab and you do all these assays that I've mentioned. And then PhD, and I supervised several PhD students as a postdoc, but now I supervise myself 2 PhD students. One of them is working on the modeling of DNMT3, which works with basically epigenetics. So we're trying to develop a model to predict whether the DNA is going to be methylated or not. And then there is another student that's working on CML, which is a biomarker for CML. So collaborators. So as you see here, you have Professor Patrick Ayres. Dominic's now at Track, so he has his own group, but we continue to work together. And then John was a PhD student in Professor Patrick Ayres' lab, and me and Dom, we trained him, and he's excellent, and we continue to collaborate and work together. And then in the same group, we have Leonard. So Leonard, he runs the omics, so he's basically the person that helps me to do all the protein identification, so I can isolate protein and then send it to him to analyze if my isolation is pure or not. That means like pull downs and many other biochemical assays. And then Professor Claire is the director of the center, who supports me with more of the logistical side of the experiments. We have Lorna. She's an artist. She's really not a cell biologist because she does imaging, but these are the type of imagings she can get with her microscope. I do a lot of imaging, but she's really, really amazing with all the imagings that she obtained. And then Neil is the one that does molecular docking, and he's the director of chemistry, and he's supporting us with all the modeling and all the papers we're working right now. And then there's Wang. Wang is an amazing electrophysiologist, and he works with mechanosensory receptors and then also with TRP channels. And because there are some projects that I'm really interested looking into calcium and oxidative stress. So we are collaborating for that project as well. James Austin he runs a technique that is really beautiful. It's called mass cytoff. So it's similar to running a flow cytometry, but instead of having an antibody that labels the antibodies at the cell surface, you have metals. So you can throw the cells in a mass spec and then identify the different metals, and that will give you a pattern of which receptor will be at the surface or not, which is the graph that he has that I added on top of the photo. Then Professor Mike Biavacci, who was a professor at the Univalley at the time I was there, and now she's at UFSC. I think most of you may know her, and we still collaborate. It's been years, and just last year we actually won an award of the UKRI, and then we've been developing a project that is almost ready to go, and hopefully will be beautiful. And then Juicy helps me with the data analysis, because I do data analysis, but then eventually when you get to the level that you need to analyze a lot of data, I rely on her because I can't keep up with all the codes that keep changing, and I don't have the time, and she's amazing. She can do that better than me. Fiona is a PhD student who I trained. She's currently in Professor McEwan's lab, and she's basically my hands in the lab. So she helps me a lot. Everything I do, I wouldn't be able to do without her, because she grows the cells for me. She looks after them. She's there for any emergency. And I must thank her so much for that. She's just published another paper this week, so she must be very happy. And this is Professor Klein too. He has the same interest as I do, which is redox and GST-Pi. So throughout my PhD, we developed a good collaboration, and then we participated in the same groups and same societies. And then he invited me to write this chapter book, which I think summarizes a lot of what I do, because it talks about the redox. It talks about how I explore cysteines for all my studies, and then it focuses on protein kinases and many of the aspects that I think are very important and are not as well studied. And I think altogether, this is really important for the whole pathology, not just acute myeloid leukemia, but leukemia in general. And I'm also organizing this meeting, which is going to be next year. It's going to be in UK. It's an international meeting. You'll be meeting people from all over the world that works with lipids and proteins. So basically protein lipidation. And we're trying to make it more translational. So if any of you work with lipids and have an interest to participate, let me know. There will be bursaries available. And if you're a member of the society, that will help. And it's really not expensive, and it's a fantastic group. And I think you would be welcome to join. And this image is a person. She's an artist. She's a painter. And she painted this as our logo, because now we are trying to engage into arts and trying to bring all of this, especially to explore students with special needs that love science, but perhaps they're not as good as writing long essays or being able to concentrate for long hours, but they have different talents and different skills, and they can bring some different input to our research. And then I think here I'm just highlighting how I think we can collaborate, because I understand you were very great at obtaining different molecular from different plant extracts. But now I've learned that you have several other skills as well that we can talk about. So that's why I left it open for debate. And before we start, I just wanted to really thank every company that or funding agency that makes the science possible. And these are them. And thank you all. It's been a pleasure to be here, and I wanted to thank you for inviting me.
Thank you very much, Professor Marensi. I think that we have a lot of things to discuss, actually, because we can highlight that there are some very interesting connections. I don't know if anyone wants to start. I should start. I don't know. Maybe Dr. Marensi, first you can maybe start your discussion. What I noticed that you are somehow interested in maybe some compounds or extracts to evaluate. Is that correct?
Yeah, I really appreciate it. So the work, for example, that I'm doing with Mike is I have this, it's like a mini library of molecules. And then what you do, you run some prediction to see which are the protein targets. And then I've done some in vitro assays because I have interest in protein kinases. So
Okay.
there are over 500 kinases because I have, of course, every technique has a limitation. I managed to do it with 146 kinases, but I was lucky to find some of like matches. And then I have some experiments in vitro that confirm those results. So now when I mentioned that I'm trying to work a way of animal models, Vinicius, please, nothing personal. I'm quite interested in zebrafish. We are developing this protocol where we can look, because kinase inhibitors, they give a lot of side effects in terms of like arrhythmia. Not really known how it works. And then what we would do as a toxicological assay, like to validate in vivo, would be to look at the heartbeat in zebrafish embryos, which will be a very first step that would do a lot of triage before we get to like higher animals like mouse and then so on. Depending on the place, we use different animals. But this is one of the things. And then another thing is because we can track developmental issues with those inhibitors, because some of them we all know can be quite toxic, and especially because they're quite hydrophobic. So this is like the step we're at in terms of natural products. And then there was, I'm trying to remember, I've seen so many interesting things. I was quite curious about, I think it was Nadia's project with the GI, where you mimic gut.
Is Thaisi?
Was it easy? I'm so sorry.
I think so. It's related to the gut experiments.
There was an in vitro experiment to look at the GI.
Nara, I think it was Thaisi.
Like the leading part of the stomach.
I will try to share the screen again. Let's just see here. Let me see. Let me check here. Mobility, chemotherapy, assessment vascular, assessment intestinal, mobility in vivo and ex vivo. But in animal models or in vitro?
In vitro.
Yeah, I'm trying to find here, but
isn't that okay. Ex vivo?
Is it ex vivo?
Oh yes, so. Yeah, intestinal mucositis in vivo and in vitro models. Yeah, but I don't know. I think I don't know if Thais is here now.
Oh, that's okay. No.
Yeah, but I think I can check with her later, because she works with this part of gut and everything. So I can put her in contact with you.
Okay, thank you. Right. And then another thing I'm trying to remember. I should have a pen here to write this down. But Professor Tania had an interesting lycopene.
Yeah.
And it's a substrate of GST pi. So I thought it was very interesting because I worked at the very molecular level of GST pi and how the substrates bind to it, because my experiment that I'm running right now is to see how lipids will modulate the binding site and change it and help or not. Because one of the experiments that I haven't published, but I'm continuing the project right now, is because when I extract the protein from bacteria and run an assay, if I include lipids, it shuts down the protein. So there's no enzymatic activity. Whereas if I extract the protein from mammalian cells, the activity is enhanced, which means that it's about, because the bacteria doesn't have an ER and doesn't have all those, doesn't come with all the components like lipids and sugars that will be available. And then there is a good chance that this is what is modulating the binding site, where there's, so we don't really know what it is. And that's where the modeling that I really want to do is try to predict, knowing where the fatty acids bind, how it would modulate the binding of substrates, where the ones we know the crystal structure is. And then the lycopene has a long chain that's similar to a fatty acid. So, and being a substrate, I don't know, is there anything known about anything with eyes in your work? Retina?
In fact, we are trying to obtain the enriched lycopene extracts, but I think next year we will have some samples, because we are optimizing the extraction, and we aiming to study some application of this extract. Could be for nutritional application or for cosmetic application. So if you are interested, we can send you some samples to test in your lab.
Yeah, that would be great. Thank you.
Yeah, and also related to the natural products, as you mentioned, of course, because I'm not sure with Professor Biavati from UFSC, you evaluate the isolated compounds. Is that correct? Yeah. Okay. Because in our case, of course, we have different isolated compounds in our in-house library, but in my case, for example, since I work with bioinformatics, I usually have to perform the assay using the struct or the fraction or etc. And how compatible are your screening assays to these complex mixtures? Because I know that there are some things in the structs or fractions that can hamper somehow this analysis. For example, tannins. Tannins are well known responsible for the precipitation of proteins, so it would hamper the assay using an isolated protein. So how do you see this as a possibility to work with complex mixtures? I know that usually you do not like to work with complex mixtures. You want the pure compound.
No, they're isolated molecules with a well-established molecular weight.
Okay, so yeah, you work with the isolated compounds.
Yeah.
Yeah.
Yeah,
okay.
because we need to know the structure to compare. So we compare the structure with whatever has been predicted for proteins that have like non-protein structure bound to. Usually, it's an inhibitor. So the software will predict how likely that molecule will bind to the protein. So it actually has to be very well defined. Yeah, it's all in silico. But then, because even though it's really nice, we need to validate. So what we do is to validate in vitro, and I try to run big assays, trying to screen for all of them. And then later on, now I would like to test these proteins, especially because they're on the stress kinase pathway that I work with. I would really like to get just those specific proteins and then get like accurate data for binding and all of this.
Okay. Yeah, I see that. We have some compounds that, of course, kinases play a role in different diseases, and but I know that there are some kinases related to Alzheimer's disease, and we have some compounds that we obtain that we already demonstrate some multifunctional profile. Targeting serotonin receptor, targeting well, at least reducing proteotoxicity related to amyloid plaques and alpha-synuclein. So this is something that we might maybe take a broader aspect regarding different targets. So maybe I can share with you some of these compounds and some of the publications later, and we can further discuss regarding these different approaches and if it's worth to go for these predictions. You know?
Yeah, that sounds interesting. I think we need to meet because you address to like at least 3 complex subjects. So one is like knowing the kinase and where they are, and then if you get into the amyloid disease, then you get a lot of ER involved. Then that means you get into the kinase there would be inside the ER, and then so there are like few things that might get involved, and the ER stress and all of these, I guess, which is really interesting. So, but that's why I think we might have to need to meet and then try to identify which aspect in detail would be explored. Yeah, no, it's quite nice.
That's great. So yeah, I will arrange some material and I can send you. I have your email and I can send you later. Perfect.
Yeah, I'm quite flexible, August.
Okay, great. Yeah.
Yeah,
of course. Go on.
Thank you very much for your presentation, Vanessa. I'm very pleased to meet you. And thanks for acknowledging that Calgary is better than Edmonton. It's just a joke. I love Edmonton, actually. You know, the only problem is a little bit further away from the Rockies, right? But it's a beautiful town. I was there in December, and I love it. So another thing that I wish to comment is that I totally share, despite we make a rational use of animal models in our research, I totally agree with you, and I think that we should stay as much away as possible in terms of the animal research. But unfortunately, there are some mechanisms for some diseases that still demand the in vivo organism. And I just would like to acknowledge your thoughts and tell you that I also share the same thoughts and ask you if one day you are able to develop, or if you know anybody who can develop a model of anxiety and depression and a way of study comorbidities in cells, I will be very pleased to introduce that to my research program, because as an animal lover, I'm also, willing to, and I think that there's the necessity to use as less animal as possible and to minimize the use of those special little ones.
Yeah.
Thank you very much.
It's great. No, but it's okay. I think for example, I wasn't even thinking much about neuroscience. I was thinking more about hormones.
Yes.
It's just really hard because you get muscles that talk to your pancreas, that talk to your heart, that talk to your whole body. So it's just
very hard.
Not feasible to do in cells.
The same thing as when you're studying brain connections, right? Like specific populations of neurons that project from one structure to the other. And it's great that we have nowadays optogenetics that we definitely can apply initially to cell cultures and to brain slices, that we can, with one mouse, make like hundreds of slices and minimize the use, and just later on apply to animal models, right?
Yeah.
But I'm totally in the field and in the side that we need to develop more specific models, computational models, cell models, what I said is not a joke, it's actually very serious, because I really take very serious the work that I do, as well as everybody else here in this presentation. And we know that we need to minimize the use of animals, but I also need to acknowledge the structure that Univali have to produce animals with a great quality, and it's not behind of what I had in Calgary. So it's still an important tool for research, depending of the field that you are, and you just acknowledge. So, and that's very important. But definitely, I believe that in the next maybe 10 years, we are going to be using less and less progressively animals for research, and that's something that we all wish, and we all should be working hard to get to that point. So thank you very much for all your input. So it's fantastic.
Thank you. Actually, I'm working with a psychiatrist here at the University of Chester, but I didn't add because it's a baby.
Okay.
But we're collecting data. So it's a questionnaire. So we really want to get into. So how do I do it? How do I put it? We want to start drawing the difference. Well, where is schizophrenia, how it gets triggered, if anxiety there's any role. And then one of the things we were thinking, the projects we have written and we are working on, and there is a medical student that's working in this questionnaire, and data that we are going to get from the clinic is, so one of them is with the ayahuasca, because there are some groups that do it very seriously for treatment. In England, it's not ayahuasca, but mushrooms extract, basically.
Yes.
So there are trials for
That's
anxiety, for OCD, and they seem to be very excellent, really.
Yes, there's actually a big trend on this field, and this is really, really important, especially because those psychiatry diseases and neurological disease, they appear usually as comorbidities, right? Usually a patient that's schizophrenic, he has anxiety or depression or psychosis. And definitely those extracts and those plants have been shown in Canada as well, especially for obsessive compulsive disorder and post-traumatic stress disorder, like a major depression with suicide ideations. So patients have been benefiting, but a discussion that we have here is how to use them safely, right? So which patient should take and which one should not take it. And there's a discussion here in the Pint of Science on a talk that I had with Professor José. It was that try to acknowledge this research and give some information, especially to the young people that are indiscriminately taking mushrooms and ayahuasca as a recreational drug and not a therapeutical drug, and that's why we worry a little bit. So I totally agree that we needed to have serious research to tell the society and the medical field which are the potential benefits of it, because there are really good benefits on it, but also to bring this to the topic and to the discussion that this is not for everyone, and that should be under the prescription and followed by a psychiatrist, as you mentioned. So this is something really, really, really interesting.
Yeah. So this project has this clinical aspect, which will be collecting data, and then there will be the aspect, because there is this bridge between anxiety and the schizophrenia, which might be related to the sharing receptors or something like this. No one really knows. And then some biomarkers at the serum biomarkers, because it's ethically, it's easy to get ethical approval for those. And then finally, in terms of studying the extract itself, so that's where also the chemists there may come. So we don't really have anyone set to do that part. So if anyone wanted to do the fractionation and the extraction, that maybe could be something that we could think of. Well, as I said, this is a baby step. It's just in the paper. It's getting ethical approval. We're sending out the questionnaires. We haven't collected any data yet.
I'm so sorry. They are sending us message that we should finish actually our meeting.
Okay.
I'm sorry that we had a short time to discuss, but in the end of the day, we can have further meeting to get deeper in each point we highlighted now. And of course, we might have other connections as synthesis and other professors who were not able to attend today. But unfortunately, we must get to an end. Dr. Marensi, first of all, we are very grateful for your presence and for your very nice presentation on very nice topics. Actually, we were aside of the discussion here, we are actually discussing with Nara and Tania that there are so many opportunities for collaboration that actually half an hour or even 1 hour would be not enough. So what we should do from our side is to take in your message home, and we'll think about some connections as we discussed, as other we didn't discuss, but we can think about. And we can send you the material, the presentation we can share with you, and then you can share with your group if you want. So, and then we can have maybe another meeting, another day, more relaxed with no deadline to finish the presentation.
Yeah.
Is that okay for you?
Yeah, I'm happy.
Okay.
And as I said, August is being quite flexible because for us it's summer break.
Yeah.
So happy to meet and anytime. And yeah, then I will take notes and I'll remember everything.
Okay. Yeah. Okay, so
it's been
I'm
a pleasure.
also very...
Thank you so much, and I loved every single research project presented.
Great. I'm also very grateful for my colleagues and also for the people attending this meeting. And I hope to see you maybe later in the afternoon in the Science Day event. So thank you very much to you all.
Thank you.
And
Thank you. Bye.
See you soon.
See you. Take care. Thank you very much.
Thank you.
Thank you. Bye-bye.
Bye-bye.
Bye.